So for the second video, we will discuss about fluids composition and movement. So the ionic and chemical composition in body fluid, they are highly variable depending on the parts of the body, organs, or function. So when we talk about composition, we talk about sodium, potassium, calcium, phosphate. All of these are ionic composition in body fluids, and whether they are in extracellular or intracellular or in the blood vessel, they have differences in the composition of these ions. So in the extracellular fluid, they are relatively high in sodium if we compare to intracellular. They are also high in chloride and also bicarbonate. So in ECF, you know, you have blood plasma and your interstitial fluid. So the difference between blood plasma and interstitial fluid composition are usually in the presence of protein and ions. Blood plasma, they have protein and ions. Interstitial fluid, they don't have protein and ion. For intracellular fluid, they have relatively high potassium when we compare to extracellular fluid, and also high in phosphate. Another thing that you need to note, intracellular fluid, they also have protein and ion. So the only space between the three main compartments that do not have protein and ions are only the interstitial fluid. So you look at this picture, so this is the two extracellular fluid composition, and this is your intracellular fluid. So you can see for plasma and interstitial fluid, they are all the same except for this protein, where you do not have protein in your interstitial fluid. When we check intracellular fluid and compare it to extracellular fluid, you can see that they have higher in potassium, phosphate, and as you can see, they also have protein and ion. So you have protein and ion in plasma and intracellular fluid, but not in your interstitial fluid. So those are the differences in the ionic composition. So all of these ionic composition with other solutes, they all have their osmotic value. And then they will form this value, this is what we call plasma osmolarity. So plasma osmolarity is something that we need to know because it usually helps us determine the patient body fluid status. So whether they're dehydrated, usually the plasma osmolarity will increase. And if they have excessive fluid, then usually the plasma osmolarity will decrease. Usually, although there are other cases as well. So the value of plasma osmolarity is usually between 275 and 295. So as you can see here, we have this corrected plasma osmolarity value. So there are a few simplified formula that can use to measure plasma osmolarity. So this is more on clinical settings where you don't have all the value, because you can see here, all of these substance, all of these contribute to plasma osmolarity. They are all counted to the final value here. But in clinical settings or in more practical setting, if you want to approximate the value of plasma osmolarity, you don't have all of this value. So there are a few formulas that have been developed that allow us to approximate the osmolarity when we only know few of the value. So as you can see here, formula 1, you only need to know sodium, glucose, and urea. Formula 2 is quite extensive. Like you need your sodium, chloride, bicarb, potassium, glucose, urea, and you need to plus 10. The third one is quite simple, whether you just have 2.1 and then you times with sodium. So this all, if you take a value and calculate, they will be slightly different from each other, but it's good enough for us to approximate osmolarity if we only have this value. Of course, this is not the best way, but it's allowed us to approximate the value. So we talk about the composition, and then we will talk about movement of fluids, because all of the spaces, the interstitial, the blood plasma, and also the intracellular, they are all connected to each other. And you know, through the membrane, the fluid can go into other spaces. So these movements are important in developing equilibrium. Changes in fluid osmolarity will cause the fluid to move accordingly to adjust the osmolarity back to ideal range. So if we have changes in osmolarity in one part of the compartment, the fluid will move accordingly to adjust, to adapt, so that it will go back into homeostasis. So the classic example, I think most of you have heard this, where you have the red blood cell when they are surrounded by solution concentration that are different in the osmolarity. And at bigger scale, you can see this is also occur between the compartment, mainly between your extracellular and intracellular fluid. So this is the example of the red blood cell. So the red blood cell, when they are surrounded by isotonic solution, then the movement of fluid exiting the cell, that means from ICF to ECF, they have normal movement. So equal numbers going out equals numbers going in. But if the surroundings solution is hypotonic, you have more fluid entering from the ECF to the ICF. And the opposite will occur if you have hypertonic solutions surrounding the cell, where you have the movement of fluid from ICF into ECF. So this is just a movement from one cell. So if we go zoom out into the bigger view, then you have the movement between your intracellular fluid compartment and your extracellular fluid compartment. So to help understanding this, when we have different conditions that affect fluid concentration, we can use is what we call a Darrow-Yannet diagram. So Darrow-Yannet diagram is very simple. So this is the Darrow-Yannet diagram. So this is your Darrow-Yannet diagram. So you can see here, you have the osmolarity for the y-axis. For the x-axis, you have the volume of fluid. And these two parts, these are your intracellular fluid and your extracellular fluid. So you can see the total here is 42 roughly, that's what we talked before, and you have 28 and you have 14 here. So this is the Darrow-Yannet diagram and how they can help us in understanding fluid movement between ICF and ECF. Then you see here. So first, you determine whether there is a fluid loss or fluid gain. So if you have diarrhea, so that means you have fluid loss, or you have excessive sweating, so that is fluid loss. Fluid gain, usually if you somehow drink excessive water, or the more common one is when you give IV infusion in the hospital. So IV infusion, if you give excessive IV infusion, then you have excessive fluid gain. So fluid gain naturally, or fluid loss naturally, will affect the x-axis because this is the volume of the fluid. So the second one, you determine what type of fluid loss or fluid gain that is happened. So the type is, what is it? Isotonic, hypertonic, or hypotonic. So this will determine the changes in the y-axis. If it's isotonic, then there is no change in osmolarity. If it is hypertonic, then usually it will increase in osmolarity, and if it's hypotonic gain, then usually it will decrease in osmolarity. So when you know these two, then you must determine how the extracellular fluid will be affected. So you must remember, extracellular fluid is the first that is affected. Always the extracellular fluid. So determine how 1 and 2, whether it's fluid loss or fluid gain, and the type of the fluid loss or gain, how it affects ECF, volume, and osmolarity. And then after that, then only the ECF will adapt to the changes that occur in ECF. So this is how we use Darrow-Yannet diagram. So if you can see here, this is example for different types of fluid gain. So you can see this is normal state. When you just add either isotonic, sodium chloride, you can see it only at the volume. There is no change in the osmolarity. But you add hypotonic solution, then you can see you have this change in volume in ICF and in ECF and ICF, and you have decrease in osmolarity. If you add hypertonic solution, then you can see there is increase in your osmolarity. And you also have increase in volume, but the volume only increase in your ECF because the fluid in ICF will go into ECF because ECF is hypertonic. So this is how the Darrow-Yannet diagram help us understand the movement of fluid. So you must know how to use the Darrow-Yannet diagram to explain the movement of water following this condition. So hypotonic water gain, so such as excessive fast saline infusion or excessive drinking water. Isotonic water gain, such as excessive normal saline infusion, or hypertonic water gain, such as 3% and 5% saline infusion. And also you need to know the water loss, how water loss also can be explained using the Darrow-Yannet diagram. So hypotonic water loss, the disease that can cause is diabetes insipidus. And then you have excessive fever or excessive sweating. Hypotonic water loss such as diarrhea or vomiting, or hypertonic water loss such as the use of diuretic medication or Addison disease. So we will discuss the types of IV infusion and the use of Darrow-Yannet diagram further in our SGD. So if this looks like quite complicated for you, don't need to worry. We will discuss in more detail during SGD. So there is also a dynamic that occur, the movement within the extracellular fluid, so which is between the blood vessel and the interstitial space. So the movement between these two spaces, they move according to what they call the Starling Forces principle. So there are 4 Starling forces that you need to know. So they are the capillary hydrostatic pressure, the capillary plasma oncotic pressure, the interstitial hydrostatic pressure, and the interstitial oncotic pressure. So the first two are in your blood vessel. Capillary, as you know, is the one type of our blood vessel. And this one, the interstitial, is in your interstitial space. Interstitial space or interstitial fluid. So hydrostatic pressure will push fluid out of the compartment that they are in. So that means capillary hydrostatic pressure will push the fluid out of the capillary. Hydrostatic oncotic pressure is different. They will pull the fluid into the compartment they are in. This is because they have proteins there. So this is how the fluid exchange occur between your blood vessels. So this is your blood vessel, the capillary. So this is your blood vessel. And the fluid here is your plasma. And this is your interstitial space where you have your interstitial fluid. So the capillary hydrostatic pressure will push fluid out, while the interstitial fluid oncotic pressure will pull the fluid into the interstitial space from the blood vessel. So you have these two pulling fluid into the interstitial space, and then you have your interstitial fluid hydrostatic pressure pushing the fluid into the blood vessel, and the plasma oncotic pressure that pull the fluid from your interstitial fluid into your blood vessel. So the dynamic of these forces allow for the filtration and absorption to occur between the blood vessel and interstitial space. So this is how you send your nutrient and everything. This is how they happen, and how you remove the toxin from the blood, and the waste from the blood. You want to remove, you need to exit the blood vessel. This is how they happen. So that is the end for the second video. For the third video, we will talk about dehydration, which is quite common when you have imbalance of fluid gain and fluid loss. Thank you.